Lake Victoria is the largest lake by area on the African continent and is a key resource for the 45 million people who live in the Lake Victoria Basin. The water of the lake is supplied by direct rainfall and by numerous streams and rivers that drain its vast catchment. However, much of the nitrogen entering the lake is via N2 fixation by the phytoplankton. The first measurements of nitrogen fixation in Lake Victoria indicated that more nitrogen may enter the lake via this mechanism than via river and atmospheric inputs. And recent molecular studies have drawn attention to blooms of nitrogen-fixing cyanobacteria in the lake. The nitrogen cycle is a complex set of biogeochemical processes in which nitrogen is converted among its various forms and redox states. In addition to N2 fixation, the many transformations include ammonification, nitrification, denitrification, and anaerobic ammonium oxidation. Nitrogen is a major constituent of all plants, animals, and microbes, and is therefore a major nutrient that affects lake and river productivity. The main forms of nitrogen in fresh waters are ammonium, molecular nitrogen, nitrite, nitrate, dissolved organic compounds, and particulate nitrogen. Dissolved organic nitrogen often constitutes over 50% of the total soluble nitrogen in fresh waters. And over half of this DON occurs as amino nitrogen forms, mostly as polypeptides and complex nitrogen compounds. Simpler forms, such as urea and dissolved free amino acids, are rapidly utilized by bacteria and algae, and therefore have low concentrations and fast turnover rates. Particulate nitrogen comprises plankton as well as non-living detrital materials. Large amounts of particulate nitrogen can be transported by rivers, especially during flood events that erode and mobilize soils across the catchment and from the riverbanks. These negatively charged soil particles adsorb ammonium, NH4+ ions, which may be subsequently desorbed and exchanged for sodium ions when the particles enter the sea. Dr. Jing Lu is a research fellow at the Australian Rivers Institute at Griffith University in Brisbane, Queensland. Her research interests include nutrient transformation and transport processes, and the application of ecological engineering approaches to restore aquatic ecosystems and their catchments. Here she describes the importance of particle size for nitrogen transport into downstream coastal marine waters.
During the riverine transport of eroded soil from upstream to downstream, larger soil particles may settle along the way, but fine sediments are likely to be carried long distances by the floodwater to the sea. These cause extensive sediment plumes that can be visible in satellite images. Fine sediments are also enriched in both nitrogen and phosphorus compared to their parent soil with larger soil particles. This enrichment poses a higher risk of nitrogen desorption and continuous release when the sediments reach to the coastal water and settle in the bay areas.
The concentrations and depth distribution of the different forms of nitrogen reflect the balance of microbial production and loss processes, in turn affected by oxygen supply. There are large differences in these vertical patterns between oligotrophic and eutrophic lakes. Ammonium concentrations are usually close to detection limits in aerobic waters because of utilization by phytoplankton in the photic zone. Additionally, bacterial nitrification occurs in aerobic waters, resulting in ammonium being oxidized to nitrite and nitrate. When the hypolimnion of a eutrophic lake becomes anaerobic, bacterial nitrification of ammonium ceases. The oxidized microzone at the sediment-water interface is also lost, which reduces the adsorptive capacity of the sediments for ammonium. A marked increase in the release of ammonium from the sediments then occurs, and as a result, the ammonium concentrations of the hypolimnion increase. Nitrate is the most common form of inorganic nitrogen entering lakes and rivers via surface runoff, inflows, groundwater, and precipitation. Nitrate is also produced within aquatic ecosystems by nitrification, and is an important oxidant for many biogeochemical reactions within the ecosystem. Nitrification usually takes place in two stages. Firstly, ammonium oxidizing archaea and bacteria oxidize ammonium to nitrite. And then secondly, nitrite oxidizing bacteria convert the nitrite to nitrate. Additionally, complete ammonium oxidizers have been discovered, and these can oxidize ammonium all the way through to nitrate. Nitrification consumes large amounts of oxygen, 2 moles of O2 for each mole of N, and can therefore be an important contributor to oxygen deficits in aquatic ecosystems. Nitrate is taken up by the phytoplankton and reduced to ammonium and amino acids inside the cells. This reduced nitrogen is subsequently released as ammonium during cell death and decomposition. Bacterial denitrification is the biochemical reduction of oxidized nitrogen forms coupled with the oxidation of organic matter. Denitrification occurs in anaerobic environments, such as in the hypolimnia of eutrophic lakes and in anoxic sediments. Dissimilatory nitrate reduction to ammonium also contributes to a nitrate removal, but its importance relative to denitrification varies among aquatic ecosystems. The processes of nitrogen cycling in rivers and streams are similar to those of in lakes, and are influenced by bacterial, fungal, and algal metabolism. Nitrification and denitrification often function simultaneously and reciprocally in running water sediments, where many microzones of steep redox gradients occur in the hyporheic zone of the streambed. In running waters, nitrogen is used repeatedly as it passes downstream. The rate of utilization and release depends on physical and biological retentiveness, largely by the microbiota attached to the streambed. The average distance a nutrient atom travels downstream during one cycle through the water, biotic, and substrate compartments is referred to as the spiraling length. In lakes and rivers, molecular N2 fixation occurs by certain taxa of cyanobacteria and heterotrophic bacteria, and may be significant in some lakes under conditions of severe inorganic nitrogen depletion. This and other biogeochemical processes are now being tracked in the aquatic environment with molecular methods. Dr. Brittany Zepernick is an incoming assistant professor in the Department of Biology and Marine Biology at the University of North Carolina, Wilmington. Her research is at the intersection of harmful algal blooms, climate change, and the freshwater marine continuum, and she applies molecular and bioinformatic approaches to investigate the ecophysiological drivers of harmful algal blooms. Here she describes a collaborative study by an international consortium of researchers at Lake Victoria.
Lake Victoria is prone to toxigenic, cyanobacterial, harmful algal blooms, which have various health and socioeconomic consequences for those living along its shoreline, as shown most recently by Julia Abuya at the Kenya Marine and Fisheries Research Institute. My colleagues and I recently investigated the phytoplankton communities involved in Lake Victoria's nitrogen cycle using a variety of molecular methods, which serve as a powerful set of tools to track community composition and function in aquatic systems. In particular, we use metagenomics, the isolation and subsequent extraction of DNA, which can tell us who is there at the time we sampled, and also what biological processes these organisms are capable of performing. In other words, this can also be referred to as an organism's genomic potential. When we dove into the metagenomic libraries to look at the relative abundance of cyanobacteria, we expected to find the cyanobacterial community dominated by Microcystis, given the various historical reports of these microcystis blooms in the region. Instead, what we found was an abundance of the nitrogen-fixing cyanobacterium dolichospermum, which contributed up to 75% of the total community at some sample sites. This was unexpected, but does corroborate prior reports estimating biological nitrogen fixation to be the largest input of fixed nitrogen in Victoria. When we looked at the relative abundance of nitrogenase genes across our sample sites and who these genes belong to, we confirmed that dolichospermum was indeed the dominant nitrogen fixer out of the cyanobacterial community and also the prokaryotic community broadly, with dolichospermum blooms suggested as a result to be nitrogen-fixing hotspots in Homa Bay and other sample sites near large urban epicenters.
From pre-industrial times to the present, there has been a large increase in the loading of inorganic nitrogen to lakes, rivers, and downstream coastal environments. This has resulted from fertilizer and manure applications, nitrogen fixation by legume crops, sewage and other wastewater discharges, and industrial emissions to the atmosphere. These increased nitrogen loads to waterways can stimulate noxious algal blooms, reduce diversity, and cause the loss of macrophyte communities. These effects can also reduce the quantity and quality of food for aquatic animals. Dr. Michele Burford is a professor in the Australian Rivers Institute at Griffith University. Her research on water quality takes a catchment-to-coast approach and aims to inform nutrient management strategies. Her work has encompassed many aspects of the aquatic nitrogen cycle, including the ecology of N2-fixing cyanobacteria, nitrogen budgets for aquaculture, organic N uptake by phytoplankton, and nitrogen transport processes to the sea. Here she describes the need to consider nitrogen as well as phosphorus control to protect and restore inland and coastal waters.
There is increasing evidence that controlling phosphorus alone is not sufficient to manage algal productivity in our lakes and reservoirs. We need to think about nitrogen too. There are nitrogen-fixing cyanobacteria which can bring nitrogen into the water from the air, but they're not sufficient to cause the algal blooms that we can see in our system. In fact, what we need to do is to be managing for both, because despite the differences that we see across terrestrial, marine and freshwater systems, nitrogen plus phosphorus limitation occurs in many places around the world. So dual management of nitrogen and phosphorus is critical to ensuring that we manage our algal blooms more effectively in the future.
This improved nutrient control requires attention to industrial, domestic, and agricultural sources, including better management of farm fertilizer applications. Climate change can also impact nitrogen budgets and transformations in freshwater ecosystems. For example, warmer temperatures increase enzyme activities and metabolic rates, which in turn affect oxygen dynamics and rates of microbial nitrogen transformation. Climate change is also expected to intensify the hydrologic cycle, including via more frequent and intense flood events, deepening and lengthening of dry periods, altered snow accumulation and melt, and changes in evapotranspiration. These impacts, in turn, will affect rates of microbial activities throughout the nitrogen cycle.